Hi, my name is Kelly Ramirez. I'm a postdoc at the Netherlands Institute of Ecology. And today I'm going to talk about, I guess, two different things. My title may be a little bit misleading for the beginning, but I hope to convince you at the end that it is necessary to start better linking soil biodiversity data globally. And the work I'm presenting today is really a huge collaborative effort with the GSBI, NOAA at the University of Colorado, Diana Wall at Colorado State University, EcoFinders, IDIV in Leipzig, Germany, where I was last year. And the ideas that came forth from all of these projects is what I'm presenting. So, soil biodiversity. Whether we study bacteria, fungi, collembola, protists, earthworms, nematodes, we all love our soil biodiversity. And what that means is we've been collecting soil biodiversity information and data quite rapidly, especially over the last 2 decades, and it's actually been an exponential increase. We've been working hard to get a better idea of global distribution patterns, the function of soil communities, et cetera, et cetera. And it almost seems like data is raining down on us, especially if we've been using these new high-throughput sequencing techniques or other new technologies that are coming out that really help us to characterize and describe soil communities. And even though data is raining down on us, our understanding of global soil biodiversity distribution patterns arguably remains incomplete. And I think people have already started to touch on this, and I think we'll hear more about this throughout the week. We can go to our site and characterize our site well, but how does that translate globally? How does that translate across ecosystems? And I want to present to you today why I think a better linking or networking of all of the data we're producing and will produce is necessary, and a greater access to this data will be instrumental to progressing scientific research. So we can't just do our own research at our own site, we need to share our information, and in that way we can better address scientific questions. So I'm going to use this case study, a project that I worked on last year or 2 years ago, to demonstrate why it's been difficult to link soil biodiversity information and why we still don't have a good idea of distribution patterns for all the different taxonomic groups in the soil. So this study, NOAA sort of started to introduce it, and I would like to say that I did not include any photos of him sampling soil. I even put in my own because I was like, oh, I don't want to embarrass him. And then he put up like 7, so I do sample soil apparently. We went to Central Park in New York City to characterize the soil biodiversity across the park. And this project was in collaboration with the Global Soil Biodiversity Initiative, and we wanted to do this because we wanted to bring soil biodiversity to the public. We wanted people to relate to the biodiversity below ground, and what better place to do it than an iconic park in the middle of Manhattan. So we went to Central Park, a group of researchers from Yale, CSU, and UC Boulder. In 8 hours, 10 of us collected over 600 soil samples across the park. The next day, we sieved the samples, and they were distributed to look at the microbial communities below ground using high-throughput sequencing. We looked at bacterial, archaeal, and eukaryotic communities. And then we also looked at a number of soil environmental parameters to characterize those communities. Okay, so I want to use this study to demonstrate why it's difficult to bring all this soil biodiversity data together that we've been creating. First, we all know soil biodiversity is immense. Even in a place like Central Park in New York City, we found over 120,000 different bacterial and archaeal sequences or species, and over 43,000 eukaryotic species. This is all sequence data that I'm presenting here. That was over 7,500 species of bacterial and archaeal per sample, and about 1,500 species of eukaryotes per sample. So it's immense, and it's undescribed. Of all the bacterial and archaeal sequences, 84% of them did not match a database, and 92% of them of eukaryotic sequences were undescribed. And this is actually not novel. In the opening talk, we talked about, it was presented on how so much of eukaryotic below-ground diversity is undescribed, and we're seeing this in our sequence data as well. So in Central Park, a place where you think, of course it's characterized, everyone goes there, it has millions of visitors a year, we know what's below ground, we actually have not characterized all of it. So finally, soil biodiversity is complex, and we all know this, but this study was really nice and unique because we looked at not only bacteria, but also archaea and eukarya in the soil. And we have our nice taxon list. And what we found was the taxonomic and phylogenetic diversity within Central Park was actually really comparable to the biodiversity we found globally when we compared to global soil samples collected from Alaska all the way down to Antarctica. Okay, so in Central Park, we can characterize biodiversity, but we also can look at the biogeographic patterns and community ecology. So in the park, we found no relationship to geographic distance. So when you collected one sample and then went 50 meters to collect the next sample, there is no relationship in the below ground communities, in either bacterial or eukaryotic communities. Maybe unsurprisingly, pH was the best predictor of bacterial community composition, and eukaryotic communities were also could be strongly predicted by soil pH. What was quite interesting was that we found strong community-level relationships between the bacterial community and the eukaryotic community, both in correlation and network analyses and overall community composition. There was a really strong relationship that was almost the second best predictor after pH of the communities. Okay, so this picture I think is amazing because it really demonstrates why we have a hard time kind of giving the public, soil's really cool. Not only are our organisms sometimes scary, but it's really not a sexy soil. It's really not sexy. And this is the best we have. We were in Central Park in New York City, and this is this is a beautiful soil, look at what we did, and it doesn't look great. So soil biodiversity is immense, it's undescribed, and it's complex. So these factors are all kind of working against us to prevent us from bringing our information together and getting a better idea of global distribution patterns. And so last year I organized a workshop with the GSBI and IDIV in Leipzig, Germany, and brought together about 15 international scientists to discuss what can we do about that? How can we build up a framework? How can we bring together soil biodiversity data? And we discussed the many reasons for why we would want such a platform, not only the scientific research advancements, but also policy and land management advancements. If we are to cope with and mitigate climate change, for example, what information do we need to give to policymakers and land managers? If we don't have all the data and information together in one place, how can we give them the best recommendations? So we proposed a global platform to link soil biodiversity data and information. And actually this week, we organized another small workshop, a think group, to start discussing how the logistics of such a platform, because it's really not, it maybe kind of sounds broad and it covers all the different disciplines out here in this room, all the different types of data. So we need people to work together, all sit together and discuss how we may go about this. So a global platform, we have our soil sample. With that, we collect a number of different types of data, from location and biome type and habitat to a list of species may be found in the soil, phylogenetic relationships, and a number of community metrics and environmental parameters. So we have this long list of data that can go along with our soil sample. And each person may be collecting that information differently, each researcher, and each discipline may be collecting that information differently. So it's not like this simple thing, oh, just throw it all in one spreadsheet. So this is almost where we started, but really let's take a step back. We don't need to panic. We can think about this in a simple way. What kind of data is out there? We have species level data, and that can range from traditional morphological descriptions to sequence data. We have environmental data, and we have functional data, such as traits or the metagenomic sequencing data that Noah presented. So how can we bring that all together under one platform? And we recognize that putting together such a platform will require time, effort, and resources. But as we go along this axis, our knowledge gained will increase. It will become a more valuable resource. and we'll gain more knowledge as we move along. So what are the first steps? First, we need to discover where our data is stored. It's possible that a lot of data is not being shared anywhere, it's not digitally accessible, or we just really don't want to share it. So that's one place. However, a lot of soil biodiversity information is shared. It's accessible digitally, or maybe the framework is set up to store your data, but you just haven't done that yet. So identifying those resources for researchers. Next, standardization, which may be sort of difficult, a difficult thing to talk about across disciplines, but what I mean by standardization is not that everyone has to collect data the same way, but clear ways to describe in which ways we collect data. And we need to describe those ways of collection for old data, set up standards for new data collection, and then we want the data that we collect and share within our own community to be applicable to others outside of our community. One easy place to start is the Global Biodiversity Information, I'm going to skip forward, facility, where they have all this biodiversity information occurrences linked through one platform, and they have over 500 million occurrences. But if you type in soil, we can't search by soil here, and we have very few representations of our soil organisms in their network. So the other reason we want standardization is for the future, for future data sets. And if we're archiving soil, like this recent publication by Craig Carey and Noah Fear pointed out, that it's important to start keeping track and archiving our soil samples so we can go back and ask questions about the biodiversity. So if we create standards now, then it'll make our life a little easier in the future. So finally, the third step would be a global platform that links all of these different sources of data and information on soil biodiversity through one global platform or portal. And this is what we have been talking about for the last 6 months and putting this together. And we're really in only these initial stages of identifying where the data is stored, and working on standardization procedures. In the future, with more resources, we hope to move towards a global platform. So what we will need is an agreed upon guidelines by the community. We need resources, and we need community buy-in. So this means not only in our own community, if you have data or you have a database that you recognize it would be beneficial to share with the rest of the community, then we can all start working together. But this is also outside of our community, and this has already been talked about. What does the public and policymakers need from us? So what questions do we need to ask now as we prepare to link our data all together? And with that, I thank you and welcome questions.
Okay, any questions? Yep. Yes, over there.
So I think this is a fantastic idea, and I'm just wondering how you intend or are beginning to think about linking in with existing databases that might have slightly different goals. I mean, there's K-Base that may never have actually gotten off the ground. There's MG-RAST, where a lot of data currently exists. Earth Microbiome Project. There's several different data initiatives from the US NSF. EarthCube is trying to link into metagenomic and other soil microbial community data as well. So there's so many initiatives that are going on. And my last question is it going to be a repository? Or would it just be a portal that would have to link in with repositories?
That's a really great question. No, our goal is not to create a repository. However, let's start with that one. If there are instances where an institution could use a repository, we want to provide guidelines for an institution to set up such a repository for their own researchers, right? We want to keep the data in the hands of the researchers and institutions, and rather provide access and a pointer to here's where that data is stored. At the very initial steps, it's really just identifying where the data is and describing that data. What is it? What kind of information did the scientists collect? Because it's always different, right? And so there's not, when I say standardization, again, going now kind of your first question, it's identifying the best way to link it. And so there will be it will be difficult, but these are problems or issues that the rest of the biodiversity community has started dealing with, and there are huge efforts to synthesize data such as museum records and bring it through into the GBIF portal or the Map of Life or the Encyclopedia of Life, collating information that's already out there and bringing it into just a new platform. So that's our first two steps. And then our final step of a portal would bring access to the data. So you could analyze the data if that's what if it was publicly available.